Hello, I'm Dr. Roberto Gonzalez Salinas, here to discuss the results of the MARS study, a Phase 2A pilot study evaluating the safety and efficacy of a novel drug, TP03, for the treatment of Demodex Blepharitis. I was the principal investigator for the study, which was conducted at the Asociación para Evitar la Ceguera in Mexico City since 2019. Demodex mites, which infest the eyelash follicles, meibomian glands, and sebaceous glands, are the most common ectoparasite found in humans. The presence of Demodex folliculorum has been implicated as a cause of chronic blepharitis, an enduring ocular inflammation that primarily involves the eyelid margin. A meta-analysis of almost 5,000 patients has shown Demodex is implicated in up to 45% of the blepharitis cases. And similar figures have been observed in regional studies worldwide. In the United States, this accounts for approximately 9 million Americans. Demodex mites can colonize the lash follicle, proliferate, and cause infestation and disease in blepharitis patients. It is thought that the mite excretions and eggs clog the follicles and create debris. This cylindrical dandruff, also known as cylindrical, are a pathognomonic sign of Demodex infestation and can be seen through a routine slit-lamp examination. This condition can often lead to misdirected or missing lashes, inflammation of another ocular tissue, blurred vision, and contact lens discontinuation. Currently, there are no FDA-approved treatments for Demodex blepharitis. Today, I'm excited to tell you about an investigational new drug that may offer a solution for patients suffering from Demodex blepharitis. TP03 is a novel topical ophthalmic drug with uniquely designed properties to most effectively and safely target the mite nervous system and thus paralyze and kill mites. It is currently being developed in a multi-dose preserved formulation. The MARS study evaluated the safety and efficacy of 0.25% TP03 for the treatment of Demodex blepharitis and constituted the first ever human use of TP03. MARS was a single-arm open-label study in which potential participants were required to have more than 10 cylindrics present on the upper lid, mild to severe lid margin erythema, and an average Demodex density, upper and lower eyelids combined number of 1.5 mites or more per lash in at least one eye. Colors were graded at a slit lamp using the grading scale shown on the slide. Participants administered 1 drop of TP03 in each eye, twice a day for 28 days, and were assessed at days 2, 7, 14, 28, 60, and 90. Efficacy was determined by the decrease in color rates and Demodex density. Safety was determined by assessing treatment-related adverse events, as well as evaluating any changes in visual acuity, intraocular pressure, and slit-lamp biomicroscopy findings. These visual results illustrate improvement in the number of color rates from baseline to day 28 for one of the 15 participants in our study. These results were typical for all participants in the study. Participants had a mean age of 69.5 years old, and 80% of them were female. The MARS study showed a statistically significant reduction in color rates as early as day 14. For the upper eyelid of the analysis eye, the mean grade at baseline was 3.07, decreasing to 0.79 by the end of the treatment period on day 28. This improvement was sustained post-treatment through the day-night visit. For mite density, again, we see a statistically significant reduction as early as day 14. At baseline, the average mite density was 2.28 mites per lash in the analysis eye, decreasing to 0.14 mites per lash after 20 days of treatment, a decrease which also sustained through the day-night visit. Therefore, in both the analysis and fellow eyes, statistically significant decreases in color rates and mite density were seen at day 14 on all subsequent follow-up visits. In this study, we observed no treatment-related adverse events, and there were no clinically significant changes in visual acuity, intraocular pressure, and slit-lamp biomicroscopy findings. At 97% of the visits, patients rated the drop as neutral to very comfortable. In conclusion, the MARS study demonstrated that the use of TP03 for 4 weeks was well tolerated and showed promising efficacy in the treatment of Demodex Blepharitis, with treatment effects persisting through 90 days. My colleagues and I are excited for a possible new therapy that targets the underlying cause of disease for nearly half of the patients with blepharitis. We thank those who have published on this topic and continue to study this condition.